Thank you very much. I'm going to be speaking about a collaboration between Georgia Tech and MIT, and I will be speaking on behalf of other authors, co-authors of this paper, Ivan Riobo, Rozga, Abowd, and Rosalind Picard. So we interact with other people in a daily basis many different times in a wide variety of settings. We are social animals and we need social interactions to explain how we feel and also to ask for help. This is very important, especially when you are young. You need to engage in social interactions to develop cognitive and motor skills. Unfortunately, when you are young, you don't have the behavioral or verbal cues like we have when we are grown up to be able to communicate and achieve successful social interactions. Therefore, this work tries to focus on measuring physiological signals, the internal state of children, to see whether we can know when they are engaged or not in a specific social interaction. I'm going to follow the same structure of the paper, so I will tell you a little bit about engagement recognition or engagement measurement in the literature, some of the specifics about the project and the data collection, and then how we go about the data processing and the classification of engagement states. So as you can imagine, there are many different ways of measuring engagement. One of the most basic ones is just through self-reports, basically asking people, how are you feeling right now? Are you engaged? Do you like this? This is a very fast and direct method, but unfortunately it's very disruptive. It requires the full cognitive attention of the person when they are responding. And this might not be really good for children when they don't have the verbal skills to be able to communicate. Another approach is to have external coders. These are usually people that know a lot about children in this case, or facial expressions in the context of facial expression analysis. And the idea is that they will look, for example, a video after the fact, and then they will be able to engage how people are feeling. The problem of this is that it's very time intensive and it's very costly. And of course, it requires the training of these expert coders. Another approach, which is complementary to the previous ones, would be measuring physiological signals. This approach involves usually putting some sensors on the body to monitor physiological signals that are related to engagement. This is less disruptive and is objective, and also it provides a quantitative measure over time. The problem with that is that it requires putting the sensors, and of course it requires instrumentation and analysis of the data. Looking into physiology, there has been many work looking at different behavioral cues in the context of engagement. So for example, people have looked at eye gaze and head orientation, head pose, to know whether people, for example, watching TV advertisements are engaged. And some other work looking at, for example, heart rate or electrodermal activity. Actually, last year, Silveira et al. Spoke about how you can measure the engagement of people watching TV by monitoring their physiological responses, electrodermal activity. A little bit more related to our work in the social engagement is interactional synchrony. This is the coordination of behaviors between individuals during social interactions. Probably you have seen this when you're speaking with someone. You scratch your head, maybe the other person starts scratching their head, and there is some mimicry on those things. That not only happens on the verbal cues or behavioral cues, but also on the physiological signals. So for example, if you are very engaged with someone, maybe your heart rate increases at the same time. This has been explored by some people. For example, Levinson at Goldman in 1983, they studied the married couples and they put a bunch of sensors, and they saw, for example, that when they were in distress, they had higher synchrony of the physiological responses. More recently, Marcy et al., they look at the electrodermal activity of 20 patient therapists, and they saw that there was more synchrony when the patient thought that the therapist was being more empathetic. Unfortunately, these approaches usually require instrumenting wired and cumbersome sensors, and they have been restricted to adults. Because children are more aware about the sensors, they get more uncomfortable, it's more difficult to get them on focus of the specific task. But now, with the development of technologies like the sensors that I'm wearing right now, you can start measuring these things more unobtrusively. However, there are some problems, and I will tell about that afterwards. So I'm going to make a brief parenthesis just to tell you a little bit more about the project. This study happens in a large multi-university NSF-funded project, which is led by Georgia Tech. And the whole idea of this project is try to develop tools to model, analyze, and visualize social and communicative behavior. This is called computational behavioral science. And here you can see more information on the website about the project, and also some of the data that you are going to see here is available for you to look at. Here you can see the experimental settings. So they have put a lot of sensors around. You can see that there are cameras, microphones. There are other sensors that you cannot see, like Kinects, and also the electrodermal activity sensors. But the idea is that we bring children into this semi-controlled setting where they engage in interactions, and then we can start monitoring how they feel and what we can tell about their behavior. As I told you, in this work, we are very interested in the internal responses, not the outside responses. Like we are going to be using the electrodermal Q sensor from Affectiva. This sensor was discontinued a couple of years ago, but it has some advantages. It's wireless and it's very comfortable. You can just put it on the wrist. And it allows for monitoring of the physiological responses, as well as the temperature and actigraphy. The measure that we are very interested here is called electrodermal activity. This is a very good indicator of arousal of the body. Usually, arousal is the amount of energy that you have, and therefore there are many things that actually influence this. So for example, if I'm thinking a lot about something, it will increase. If I'm standing or I have some movement, that also will make it increase, which adds some of the challenges to the analysis. Here you can see a small clip of how the experimental setting looks like. So we have the parent and the child coming over and then engaging in social interaction. I will let you know more about it now. But one of the things that you can see is that the sensors are on the wrists of the child and the examiner. So for the social interaction, they have developed a semi-structured interaction which involves 5 different parts. These different parts are thought to engage and elicit certain behavioral cues which are related to development. Some of these, for example, involve like greeting, saying hello, and see if the child is responding, having a book and see if the person is also looking at the book with you, or maybe having a ball and just bouncing back and forth to see if they grab it. In our specific setting, the external coder rated the engagement levels of the children, and here engagement was defined as the amount of effort required to get the child's attention. For each one of the sections, the adult basically rated from 0 to 2, 0 meaning 0 effort, so it was very easy to engage with the child, and 2 indicating that it required some effort to engage in interaction. Oops, sorry about that. So we collected information from many different sessions, and also we synchronized them. Unfortunately, we had some problems with the data collection. These are children, these are very problematic. I was telling you about some of the difficulties of measuring these things. And 23 of the sessions contain a lot of sensor artifacts. This included, for example, loose sensors, so basically the electrodes were not touching the skin, and also not enough moisture on the skin. So we end up refining the system and the protocol, like putting gel, exploring other locations and things like that. And we can tell you more about it afterwards. The final set that we're analyzing here includes 51 sessions, which balance gender for the children. And actually there were like 4 different adults that performed the protocol. And these adults, they had experience already and they had been trained to interact with these children. The average age was a bit less than 2 years. And the duration of the social interaction for the 5 parts, it was around a little bit less than 3 minutes. For the analysis, what we did was to cluster these social interactions in 2 different groups. The group that always was easy to engage, and the group that, at least for one of the parts, was a bit more difficult to engage. And there were different reasons why we performed this separation. One of them is that we wanted to analyze the electrodermal responses, and that requires a little bit of time to be able to gather like some characteristics. Like for example, the hello is only lasted for 5 seconds. And basically, the things that we wanted to measure in this specific setting was looking at the synchrony. So we need a lot of time for being able to capture those things. So in order to characterize the electrodermal activity, we follow several steps. The first one is normalizing the electrodermal responses. Different people have different frequency ranges, or actually dynamic ranges, as you can expect. Things like the amount of weight that you have, the age that you have, those influence. So we need to try to put them in the same range of values. Then we also reduce some of the noise with a filter. And then from the physiological response that we get, the electrodermal activity, we extracted two components from this. We extracted the tonic component, which is the green slow changing signal that you can see, and then the phasic component, which is the more spiky signal, which is usually associated to more specific stimulus. And by extracting these two components, we are able to get more interpretability on our findings. In order to extract the features, we look at two different sets of features. The first ones come just by looking at the physiological responses of the child. So basically, we took the whole social interaction and extracted the features that you see here, like the position of the minimum value, the position of the maximum value, the standard deviation, the area under the curve, all these to try to characterize how the physiological response is changing. But then we also look at the synchrony-based features. But basically, we took the physiological responses of the adult and the child, and then we extracted several metrics. One of the standard metrics is the Pearson's product moment correlation, which is just like the standard correlation. But we also look at canonical correlations and dynamic time warping, just to be able to model some of the specifics of social interactions. So, for example, where you are bouncing the ball back and forth, you want to see basically the physiological changes might change alternatively based on the turn taking. So we want to be able to capture those too. And then we extract some more gross metrics, like for example the means, the number of peaks, and compare the two different physiological signals. So basically we extracted individual features and synchrony features from the tonic and phasic components from these 51 couples. And then we wanted to assess the qualitative performance of these features. We address this problem as an engagement recognition using supervised learning. We use supervector machines in its linear form, and then we use tenfold cross validation for training and testing. Before I go into the details of the numbers, here you can see the average electrodermal response for the children easy to engage and the average electrodermal response for the children in the harder to engage group. And there are two things that you can quickly see. One of them is that there is an increasing pattern on the people that are harder to engage. And this is something to be expected because, as you can see, they might get frustrated over time. The adult is trying to engage them in the interaction, so there is some back and forth around there. The other thing is also the duration of the sessions of the harder to engage. They were a little bit longer because of that very reason, that the adult was trying to engage in the interaction more than the other group. So here you can see some of the results. We have all the different features and performances on the paper, but here I will show you some of the best ones for different experiments. The first round of experiments, we try to characterize one feature at a time, and these are one-dimensional features. So, for example, the average of the whole physiological response, the number of peaks, and things like that. The average performance that we obtained, or the best performance we obtained, sorry, it was 69%. It was similar performance for the individual features and the synchrony-based features, which is very interesting. Which is a little bit more interesting is that for the individual features, the tonic component, which is the slow changing signal, was more relevant than the phasic. And when we are characterizing the synchrony of the two different people, looking at the phasic component was more relevant. Here you can see that the position of the maximum value from the tonic component was the best predictor of individual features, and the number of peaks, or the difference between the number of peaks between the children and the adult, were the ones that provided better performance for the synchrony-based features. Actually, the synchrony-based features were 13% higher than traditional canonical correlation, which is the measurement that they use in the literature. And also, by extracting the tonic and phasic components, we were able to improve performance, but also to be able to understand where these differences come from. The second round of experiments, we use sequential feature selection to be able to aggregate these features and see if combinations of features actually improve the performance. And indeed, that was the case. We were able to improve around 11% of the performance by aggregating features. Again, the individual features and synchrony-based features perform more or less the same. Then the synchrony-based features, they were slightly higher, only 2%. But again, tonic and phasic components, both of them were complementary, or actually they performed more or less the same, and they were complementary. When we selected groups of features, all of them were selected more or less the same amount of times. And here you can see some of the features, like the one that we saw before, the number of peaks from the phasic components, but also some other features, like for example dynamic time warping or standard deviation, that they were not the best predictors before, but when we use them in collaboration or in combination with other features, then they have they add predictive value. So just to conclude, we evaluated 51 child-adult diets, the physiological responses of these people, and then we assessed the performance of individual and synchrony-based features from the tonic and phasic components. There are more details on the paper about the specific details of each one of the different combinations. We show some promising results, around 81% of accuracy. And some of the main benefits of this approach is that it's automatic and potentially you can scale it up. Hopefully, if you just have the sensors, you won't have to have an expert coder that provides you the ratings to be able to measure the engagement of children. The biggest challenge that we found, it was that 31% of the sessions had to be excluded. We learned a lot from that, and we can share the information. But that was something that we didn't expect at the beginning, and then we encountered it. In the future, we are going to look at the correlation of these metrics with other, for example, the head pose, head orientation that is being performed by other universities. So we are going to put them together. But the ultimate goal of this project and the whole project overall is to better quantify and understand behavior. The whole idea is that if we can quantify behavior, hopefully we can also detect delayed developmental detect developmental delays, which usually happen, for example, in children with autism. And with that, I'm just going to open for questions.